Hello everyone, welcome to my channel. So today we will be talking about the same chapter wherein we have already talked about sexual reproduction in flowering plants. So this is the next part of it. So this is the next part of it. This is the part 3 because in the first part we have discussed about the parts of flower. Then I have discussed about, in the second part, I have discussed about microspore and microsporogenesis. And now we will be talking about megasporogenesis. So this is what we will be going ahead and talking about presently. Okay. So now to go ahead and talk about what is megasporogenesis. To what is megasporogenesis? Megasporogenesis. Before that, let me talk about what is microsporogenesis because we have talked about it earlier. So, in microsporogenesis, what is happening in microsporogenesis? We are already talking about uh, uh, microspores which was available in the pollen grains, and there we were growing that microspores which was present in the sporogenous tissues that was growing into a pollen grain. So it was very small, hence the name microsporogenesis. And now we are coming to megasporogenesis, which is present in the carpel, or it is in the female or gynecium, that is which has got stigma, style, ovary and ovules. Now based on how many pistils are there in a flower, I can always segregate it into two parts. One is monocarpellary and other is multicarpellary. If it has got one carpel, I will say it is monocarpellary. And if it has got more than one carpel, I will have tell it as multicarpellary. Now, this multicarpellary, if I say, can be of two types. It can be fused and it can be free. So if it is fused, I say it's syncarpus, and if it is free, I'll say it apocarpus. So now, like if you see, this is a monocarpellary that I have. Usually I find it in hibiscus flower. So this is the one that I have extracted from hibiscus flower. So this is a monocarpellary. As you will see in a hibiscus flower, the whole flower has only one ovary. Whereas if I see in this plants which we say champak, or we have the magnolia flowers, we usually have it is multicarpellary. Whether I have this or this, both are multicarpellary, but here you see they are free. This, all the carpels are free, they are completely free. So this multicarpellary, they are having number of ovaries. The carpels are completely free of each other. Hence, I call this as apocarpus. I call this apocarpus. And here you see the carpels are fused. Hence, I say it as syncarpus. Hence, I see stated the syncarpus. Okay. So next, coming across uh, now, what is the difference between multicarpellary and monocarpellary? So, in a multicarpellary or a monocarpellary, as far as definition is concerned, I will say when gynecium consists of one carpel, we call it as monocarpellary, and multicarpellary is when the gynecium has numerous carpels, we call it multicarpellary. Number of carpels in monocarpellary it is only one, and there are numerous carpels in the multicarpellary. Examples of monocarpals are seen in family of leguminous, mango, avocado. So you have seen that there's only one seed inside, that there's only one fruit that you are able to get. So there is only one ovary that is there, hence. I can always say that is monocarpal. And they are monocarpous in nature. Now, when I talk about multicarpally, they are seen in strawberry. If you have seen a strawberry, you will have small seeds all around. So there are numerous raspberries. If you have found the raspberries, they have numerous ovules, which is ovaries, which is there. So all this are the multicarpellate. So they can be syncarpous or apocarpous. Now, as I was telling to you that this is a pistil wherein I have ovary, I have style, I have stigma. When I said apocarpus, what did I say? The all the ovaries that I have, they are completely free of each other. So they are free of each other. They are completely free. Each and every ovary has its own ovules. Hence, they are apocarpus. Okay. Now, if you see it in a pea, if I talk about a pea plant, so if you see a pea, then you will have one seed, one seed, one seed. So this is kind of seeds that you will have. Okay. Now, if I come to syncarpus, so in this case, what happens? The carpel or the ovary is fused and there are a number of ovules inside this. If you go ahead a tomato, if you find a tomato, you will find that it will have structures or cucumber. So it will have, and there will be ovules inside this. There will small seeds. So this is, if I'm talking about the flower, I'm not talking about the fruit, but I'm talking about the flower of peas, or I'm talking about the flower of tomato. So you will have apocarpus when it is free and syncarpus when it is fused. Okay. So this is about how I segregate the carpels, or how I segregate the flowers as per as the carpels which is present. Okay. So here I've given you an example. So I have the gynoecium. I have got hibiscus flower, which is monocarpellary. I have got in syncarpus, multicarpellary papaver. I have that, and mycelia, which is champak. I have it as apocarpus or free in nature. So this, now coming to what is microsporangium. So all the female gamete is present inside the microsporangium, which is the ovule that I call it. Ovule is a small structure attached to the placenta. So we have the ovule over here. So the ovule is there. I'm seeing the ovule, and it is attached. There is a placenta which is there. And in this placenta, there's a stalk which I say, which is a funicle that I say here, which is a stalk by which ovule is attached to the placenta. So here there's a placenta that I have here, and it is being attached, and this attached the stalk is known as funicle. What is hilum? Hilum is nothing but the junction between the ovule and the funicle. So where the funicle is coming and binding or getting attached to the ovule. Okay, so the funicle is coming and getting attached. So this attachment area is known as the funicle. Okay, next I will have some protective layers. These are the various protective layers I have. So they are known as integuments. I have the outer integuments and inner integuments. So this integuments has formed a small passage over here. This end I call it as the micropylar end or micropylar pole. The opposite end is the chalazal end. So now inside this I have got numerous nucellus. They are nucellus, not nucleus. They're nucellus. It's a mass of cells which is enclosed in the integuments. And these nucellus that I have, they are an abundant source of food reserve. And inside this, I have the embryo sac. So inside this, I will be having the embryo sac. Okay, so now the structure of microsporangium is clear to us. Now coming to what we call it as the microsporogenesis. Microsporogenesis is nothing but it refers to the development of megaspores. I'm talking about megasporogenesis. Microsporogenesis is when there is a formation of pollen grains from the microspore mother cells, and megasporogenesis refers to the development of megaspore from the megasporocyte or the cell which it undergoes meiosis. So this cell, the megaspore or the ovule, inside ovule, I showed you just previously, there is a megaspore mother cell which is completely at the center. So this megaspore mother cell, it undergoes meiosis. When it undergoes two consecutive meiosis, it forms four haploid megaspores. The four haploid megaspores that we have, that is seen, is then after that, the three megaspores that is being formed, they get degenerated. They get degenerated, and I find a functional single megaspore. Okay, so this is what is megasporogenesis. So once the megasporogenesis is done, I told you that three megaspores, they get degenerated. Why do they get degenerated? They get degenerated because of programmed cell death, which may be closely related with the change in the calcium content. So when the megaspore tetra decreases, the calcium content in the cell, it becomes to degenerate, and when the calcium increases in the cell, it will continue to develop into the functional megaspore. A lot of calcium is required, hence there is a reserve of calcium is there when three cells gets degenerated and I have only one functional megaspore cell that is I'm having. The megaspore at the chalazo end is functional, and three at the micropylar end that gets degenerated. Now, as I was telling you, so I have got at the micropylar end, I have one megaspore, and this megaspore, it undergoes meiosis, and there is a formation of tetrad. Okay, there is a formation of tetrad. From here, it went one meiosis. The second meiosis, it forms a tetrad. Tetrad means four cells are being formed. Okay. And around this is the nucellus, because I told you nucellus is a reserve of the food reserve which is required for the uh, meiosis to occur or me to get the megaspore tetrad. So once the megaspore tetrad is formed, I told you the three from the micropylar end, the three, it will get degenerated, and I will have finally one functional megaspore cell. So this functional megaspore cell that I have, so there I will have the division that is taking place of the nucleus alone. So the nucleus will start getting divided. So the nucleus, there is a mitotic division that has taken place. One formed two nucleus. So maybe at the chalazal end, I had only one nucleus. So now from there, I had a mitotic division which formed two. Then again another mitotic division, four, two, it was each end, and then I have another mitotic division, 4 plus 4, that went 8 nucleus I am having. So once this nucleus is formed, then what happens is 3 nucleus moves towards the chalazal end, micropylar end, and 3 nucleus towards the micropylar. So chalazal end and micropylar end, 3 nucleus would separate up and will go towards the each end, and there they will form the cell wall. So you see there is towards the antipodal, or this is the same as the chalazal end. I have got the 3 nucleus, and towards the micropylar end also I have got 3 nucleus. Now there is a formation of cell walls for all the 3 nucleus that is happening. So if I take this, 3 nucleus is there and 3 nucleus is here. So there is a formation of cell wall that is happening. So now once the cell wall is forming. Then the cell walls that forms in the chalazal end, I will call it antipodal cells. And this I have got two synergids, and one is the axil. One is the axil. So one axil and two synergids are there. So here at the chalazal end, I have got the antipodal cells. So now you see there are two free nucleus I have at the center. So I have two free nucleus. There were 8. The 3 went towards the chalazal end, 3 went towards the micropylar ends, and there are 2 more which is there in the middle, free nuclei. So this I call as 2 polar nuclei which is there. Hence, once I form this polar nuclei, okay, so all these things, now this at the micropylar end, we have filiform apparatus. So what is the use of filiform apparatus? Filiform apparatus helps to direct the vegetative cell towards the ovary. Okay, so towards the ovary. So now for fertilization, it is important. Now, this cell that I have, finally the cell that I am getting, the embryo that I am getting, the mature embryo sac, this is known as 8 nucleate 7 cells. Why is it known as 8 nucleate 7 cells? Now coming here, the megaspore. So I here what I have told, the 3 nucleate is moving up, forming 3 antipodal cells. So I have got 3 antipodal cells. Then I have got 2 synergids. I have got 1 egg cell, and this whole the embryo sac is 1 cell. So how many I have? 3 plus 1, 4, 5, 6, 7. So I have got 7 cells. And how many nucleus I have got? 3 nucleus at the top, 3 nucleus at the bottom, 6 nucleus, and 2 polar nucleus in the center. So 8 nucleates. So there are 8 nucleus, and there is formation of 7 cells. So I call this stage as 8 nucleate 7 cell. In microsporangia, what were we calling that? In microsporangia, we had vegetative and generative cells. Hence, we were calling it cell in a cell or 2 cell structure or 2 cell stage. All right. So this is the structure that I have for the megasporangium. Okay. Thank you.